Myself Dr. Gunjan, and as you can see, we'll begin our series on menstrual cycle. And in this today we'll be discussing the basic interaction of the hormones. Now before moving on to the hormonal interactions, let's first have a quick look at oogenesis. We had discussed this in detail in OBS. We'll just have a quick revision. Now what is this oogenesis? The formation of ovum, and it all begins in the intrauterine life. The germ cells, they are formed in the epiblast, and this occurs at 2 weeks, and subsequently they migrate to the yolk sac, and this migration it occurs at 3 weeks. And this was an important question. The migration of germ cell to the yolk sac occurs at what gestational age? And it is at 3 weeks. Now from the yolk sac they have to move on to the genital ridge, which basically forms the gonads. So this migration to the genital ridge, it is completed by 5 weeks. Now these germ cells, they undergo rapid mitotic division in the gonads, and now they are called as oogonia. And here they are diploid cells, so 46XX. Now most of the germ cells, they continue the rapid mitotic division, but few of them, they enter the prophase of the first meiotic division, and now they are called as primary oocytes. Even these are diploid cells, and they are 46XX. Now here in the prophase of the first meiotic division, they get arrested in the diplotene stage. Diplotene stage of the prophase, they get arrested, and they remain here in this stage till ovulation. And specifically in the diplotene stage, they get arrested at the dictate stage, the resting stage. So these primary oocytes, they get arrested at the dictate state of the diplotene stage of the prophase of the first meiotic division. And this entire statement is very important. You can get many questions from this one statement. Now this primary oocyte, it remains in this state till before ovulation. And here these primary oocytes, they get surrounded by the granulosa cells present in the cortex and theca cells present in the medulla of the ovary. And this leads to the formation of complete structure called primordial follicle. Here you can see in this image, this is our primary oocyte, and this is being surrounded by these flat cells. Here you can see here, these are the flat cells in the first row, and these are the follicular cells or the granulosa cells. And outside that you find a layer of stromal cells, and these stromal cells, they form the theca cells. And this entire structure is called a primordial follicle. So this remains as such till before ovulation. Now at puberty what happens? The hypothalamus gets activated, and now this hypothalamus, it secretes GnRH impulses, gonadotropin releasing hormone, and this gonadotropin releasing hormone, it acts on the anterior pituitary and thereby to release LH and FSH. Now this LH, which is released from the anterior pituitary, it leads to LH surge, and finally the meiosis I resumes. Again one important question, the meiosis I resumes under the effect of, and it is the LH hormone, the luteinizing hormone. Now as the meiosis I resumes and is completed, this primary oocyte now leads to the formation of secondary oocyte and the first polar body. Now as we know that this meiosis I, it is a reduction division. So in this, the diploid cells, the one with 46XX, they have become now 23X. That is division of the nuclear material, and half of the nuclear material along with the maximum cytoplasm, it comes to the secondary oocyte. Whereas the first polar body, it has the half of the nuclear material with very scanty cytoplasm. Now these secondary oocytes, they enter meiosis II. Now it is this secondary oocyte which is released at the time of ovulation. Again one important question, that the oocyte is released at which stage? And it is the secondary oocyte state. Now these secondary oocytes, they enter the meiosis II, and in this they get arrested at metaphase II, and they remain arrested in this state till before fertilization. Now once the fertilization occurs, this meiosis II resumes, and now it leads to the formation of a mature ovum and another polar body. And since it is a multiplication division, so both of them will have the haploid number of chromosomes, that is 23X. So after fertilization, it will lead to the formation of a mature ovum, which is the female pronucleus, and a polar body and will have a male pronucleus, and both of them will undergo fertilization leading to the formation of zygote. Now see, so many questions you get from this. Now here one more question that you are asked. The first polar body, it is released at the time of, and the answer is ovulation. I have already explained you that at the time of ovulation, the meiosis I resumes under the effect of LH surge, and this will lead to the formation of the secondary oocyte and the first polar body. Now the second polar body, this is formed at the time of fertilization. Now let's have a quick look at the timelines. And I have already told you that the germ cells, they appear at 2 weeks from the epiblast. Then at 3 weeks, they migrate to the yolk sac. Then by 5 weeks, they completely migrate to the genital ridge, which are the future gonads. Now in the gonads, the oogonia, it is formed at 9 weeks. Then 3 weeks later, the primary oocyte is formed, that is around 12 to 15 weeks. Then after 3 weeks, the follicle formation starts, that is around 15 to 20 weeks. Then the follicle formation completes by 22 to 24 weeks. Then again, the maximum number of follicles, they are formed at 20 weeks of intrauterine life, and that is 6 to 7 million. Again, a very favorite question. The maximum number of follicles, they are present at 20 weeks of intrauterine life, and that is 6 to 7 million. Now these follicles, they keep on undergoing atresia at the same time. So the maximum number of follicles at birth, it's just 2 million. From 7 million, it decreases to 1 to 2 million. The atresia still continues, and the maximum number of follicles at puberty, it's just 4 to 5 lakhs. And out of this, only 500 follicles, they mature in the entire lifespan, and around 1000 undergo atresia every month. Now having said this, let's move on to the hormonal interactions. And as I told you that at puberty, the hypothalamo-pituitary ovarian axis, that is the HPO axis, it becomes functional. And this hypothalamus, it releases gonadotropin releasing hormone in a pulsatile fashion. This acts on the anterior pituitary, from where the FSH and LH, they are released. And the target hormone of this FSH and LH, these are the ovaries, the gonads. Now let's see the function of FSH. And as the name suggests, follicle stimulating hormone. So the basic function of this is to stimulate the forming follicles. So they stimulate the follicles and prevent the follicles from undergoing atresia. So any follicle on which the FSH is acting, it will not undergo atresia, it will remain as such. And subsequently, the growth of the follicle will be stimulated. So the follicle will start growing. Now if you try to recall the structure of the primordial follicle that we have discussed just now, we have a primary oocyte, and this primary oocyte, it is surrounded by the granulosa cells, the flat cells, and these are the granulosa cells. Outside this, the theca cells, they are present. So this primordial follicle, it has got two types of cells, the granulosa cells and the theca cells. Now this FSH, the follicle stimulating hormone released from the anterior pituitary, it acts on the granulosa cells and leading to the release of estrogen and inhibin B. So two hormones that are released from the granulosa cells, they are the estrogen and the inhibin B. And this inhibin B, it has got a negative feedback action on the FSH. Now this is again an important question. You are asked that what inhibits FSH, and in the options you will find inhibin B. Now this estrogen, it has got multiple functions. Let us see the functions of estrogen. It has a negative feedback on FSH, thereby decreasing the FSH levels. You should remember one very basic principle, that if a hormone A leads to the production of hormone B, then this hormone B, it will always inhibit hormone A. Because once this hormone is produced in sufficient amount, it has to give a signal to the body that no more production is required, and for that it inhibits its precursor. So FSH acts on the granulosa cells to release estrogen, and this estrogen, it has a negative feedback action on the FSH, thereby decreasing the FSH levels. Now by this time, one of the follicle is selected, and that is the dominant follicle. And apart from this, all the follicles, they die because of the decrease of this FSH. I told you that this FSH, it prevents the atresia of the primordial follicle. So once the FSH levels decrease, all the follicles will die except one follicle, and that is the dominant follicle. And it is from this dominant follicle that ovulation occurs subsequently. Now apart from this estrogen, it has got a positive feedback action on the LH, negative feedback action on FSH, and positive feedback action on LH, thereby causing increase of the LH levels. Then they have another very important function, that on the uterine endometrium they cause proliferation. Proliferation of the uterine endometrium occurs, and this is the reason that in hormonal replacement therapies, HRT, that we give in cases of menopausal females, if estrogen is given alone, then it can lead to endometrial hyperplasia and subsequently endometrial cancer. So if uterus is present, estrogen should never be given alone. It should always be combined with a progesterone. Now we have discussed the functions of FSH. Now let's see the functions of LH. How does this LH act? The estrogen, it has a positive feedback action on the LH, thereby it will lead to increase in the LH levels. Now this increased LH, it will subsequently lead to LH surge. LH surge means the rise of the LH levels. And it is because of this LH surge, I told you, that resumption of Meiosis I occurs, and subsequently ovulation will occur. And for this LH surge to occur, the estrogen levels, they should remain elevated for another 24 to 48 hours. Now the second function of LH hormones is that they act on the theca cells, the outer cell layer of the primordial follicle, and here it leads to the production of androgens. Now these androgens, they diffuse into the granulosa cells, and here these androgens, they are converted to estrogen by the action of aromatase enzyme. So aromatization occurs, by which the androgen is converted to estrogen, and this occurs in the granulosa cells. Now here remember this, that this aromatization by which the androgens are converted to estrogen, apart from occurring in the granulosa cells, they also occur in the adipose tissue, the peripheral tissues. But the difference is that this estrogen produced in the ovaries, this is mainly estradiol, whereas the estrogen that is produced in the adipose tissue, it is mainly estrone. And this estrone becomes the major estrogen in cases of postmenopausal females, because in them the ovarian secretion of the estrogen, that is the estradiol, does not occur. Now coming back to the action of LH, and apart from theca cells, they also act on the granulosa cells. Now here they will cause a luteinization of the granulosa cells, and thereby leading to the release of progesterone and prostaglandins. So you see here that all the three steroid hormones, they have been formed. The estrogen was formed, the androgens are formed, and the progesterone is formed. Now for the synthesis of this estrogen in the follicular phase, two-cell, two-gonadotropin theory has been advocated, and let us see what is this. Now the basic pathway for synthesis of the estrogen is that this cholesterol, it is acted upon by the enzyme CYP17 and androgens are formed. Then under the action of the aromatase enzyme, the androgens, they are converted to estrogen. So for the formation of estrogen, two enzymes are required, CYP17 and aromatase. But there is a basic difference in the granulosa cells and the theca cells. In the granulosa cells, the CYP17 is absent and the aromatase is present, whereas in the theca cells, the CYP17 is present and the aromatase is absent. So for the synthesis of estrogen, both the granulosa cells and the theca cells are required. So in the theca cells, the cholesterol, it is acted upon by the CYP17 and the androgens are formed, and this occurs under the basic influence of the LH hormone, the luteinizing hormone. We had discussed that this luteinizing hormone, it helps in the formation of androgens in the theca cells. Now this androgen, it is diffuses into the granulosa cells, and in the granulosa cells, under the effect of the enzyme aromatase, this androgen is converted to estrogen, and this occurs under the influence of FSH, the follicle stimulating hormone. So you see here that two cells, that is the granulosa cell and the theca cells, are involved, and two different gonadotropins are involved, the LH and the FSH. So this is the two-cell, two-gonadotropin theory for the synthesis of estrogen in the follicular phase. Now the action of LH, we had already discussed that this helps in the resumption of meiosis I leading to ovulation. Then second, we saw here that it leads to the formation of androgens. Now the third, it acts on the granulosa cells, and here it causes luteinization of the granulosa cells. And from these luteinized granulosa cells, progesterone is released. So small amount of progesterone, it is released even before the ovulation. And this small amount of progesterone, it has a positive feedback effect on LH and FSH. So here you see in small concentrations, the progesterone, it has a positive feedback effect on LH and FSH. Whereas in high concentration, this progesterone will have a negative feedback effect. And subsequently, it will lead to LH surge and ovulation. So this was about the basic hormonal interactions. Now in our next class, we'll be studying the